Let's go ahead and get started. Today is Tuesday, October 26th, and we are on to the fourth section of this class, respiratory system and urinary system. And we will start with the respiratory system first. A fair amount of assignments due on this. 2 unit reviews, which you've done before. PhysioX and a Labster, which you have done before. I should say for the respiratory system, and actually something similar for the urinary system. The urinary system is going to have 2 unit reviews, a Labster and a PhysioX. So some symmetry there. There is one important, fun, but important activity I want to talk to you about. In your module, there is a control of respiration home lab. Well, you'll be doing things like holding your breath or stuff along those lines. This is a fun activity, a way to do a wet lab at home. However, there are some big, huge caveats. If you have any type of respiratory distress or respiratory disorder, COPD, asthma, any of those things, you should not participate in this activity. Do not do this activity. Do not do this activity by yourself. Either have somebody else in your family who's healthy and capable of doing the activities, read the activity, the entire exercise first to understand everything that it entails. And whether you are the guinea pig or somebody else is the guinea pig, please do not participate in this activity by yourself. Make sure you are observed, or whoever is doing it is observed while you're doing it. And if for any reason you are not capable or not comfortable doing this activity, it is not required. If you can't or are not comfortable doing the activities, then I want you to simply fill it out as a thought experiment. Write what you think would happen in these situations. Again, I'm purposely not grading this one for correctness because I don't want anybody to do anything they're not comfortable with. So again, like most homework assignments, I'm looking for time and effort, thought put into it. So if you're not capable of doing some or all of the activities, that is perfectly acceptable. Just like I said, just write what you think would happen or what your expectations based on what we talk about in class and your other activities like the PhysioX and the Labsters that you do, what you expect the results to be. All right. So again, nobody should be putting themselves in any kind of harm's way to do this activity. It's a fun activity for those who want to participate in it, but it is not required. From that standpoint, you don't actually have to do the activities. You have to fill out the handout, you have to answer the questions, but you don't actually have to do the activities to do that. It's fun if you do, but it's also perfectly okay if you don't. The other thing that I want to talk about before we get started is the last lab and lecture exams. The scores were much, much better on this. Especially in the lab exam, which I was very, very happy to see, and not surprising because it was a lot more straightforward. However, I still think there is lots of room for improvement, especially on the lecture exam. I was surprised by the number of people who seemed unprepared for the digestive pathway question, especially since I specifically told you there was going to be an essay question where you were going to be responsible for one of the pathways, digestive pathways, either of lipids or carbohydrates, or lipids, carbohydrates, proteins, or water. I specifically told you one of those 4 was going to be an essay question on the exam, and yet some people still seemed unprepared for that. So again, make sure you're attending to things that are important, things that we spend a lot of time on, or things that are on the exam. And also make sure you have details as well, specificity, and are specific and precise in describing these things. Those details can nick you points as well. All right, questions on any of that? All right, well then onward and upward. We are done with the old, let's move on to the new. As I mentioned, our goal is to talk about the respiratory system and the things that that entails. Obviously, respiratory anatomy is a part of that. The process of ventilation, and by ventilation, I mean the act of moving air into and out of our lungs. Of course, the reason we do that is to exchange gases, gases plural, And there are two places where that takes place. And then, of course, like all of our organ systems, we need to be able to control it as well. When we think of respiration in the body, respiration can mean a lot of things. One of the things that it can mean, as I mentioned, is the mechanical movement of air into and out of the lungs. And that mechanical movement of air into and out of the lungs is what we call our pulmonary ventilation. The goal of that is so that those gases, plural, can partake in external gas exchange. Now, let's think about this. Let's start first easy. What are the gases that are involved in external gas exchange? All right, excellent. Carbon dioxide is one of them. And what's the other one? There we go, oxygen. Excellent, excellent. So those are the gases exchanged. Where does this exchange take place? And when we're talking about where this is taking place, there's two ways we can think of it. We can think of it of the structures where the exchange is occurring between, or we could think of them in terms of the mediums or the substances that are exchanging the gases. So let's think of this in both ways. Pick your poison. Either tell me the mediums or the structures where this exchange is taking place.
In alveoli.
Excellent. So that would be one of the structures. We have those little grape-shaped alveoli, or the singular and alveolus, located in the lungs. And what would the other structure be then?
Pulmonary capillaries.
Excellent. Excellent. Spectacular. Now that we know the structures, let's think about the mediums or the substances that they contain. What substance or medium is contained within the lungs? What should be in your lungs?
Oxygen.
Okay, well, but those are the gases. What is containing that oxygen?
Blood.
Hopefully there's not too much blood in your lungs. You've got the right idea, though, in that should be what's inside the pulmonary capillaries. So inside the pulmonary capillaries, we should have blood. But again, I think you guys are overthinking this. What is containing the oxygen and the CO2 inside of your alveoli?
Air.
There you go. Absolutely, air. So notice this gas exchange between CO2 and oxygen, or this exchange of gases, the exchange of gases of CO2 and oxygen take place between the alveoli of the lungs and the pulmonary capillaries. Or you could think of it in terms of between the air and the blood. And of course, there's one way we can identify what's going on here in the external gas exchange, the direction. Which way does oxygen move?
Into the alveoli.
Okay, excellent. So, so oxygen in this external gas exchange, obviously the pulmonary ventilation gets oxygen here, but does oxygen go into the air or does oxygen go into the blood? Does it go from the air to the blood or from the blood to the air?
From the air to the blood.
Excellent. Or we could say that it moves from the alveolus to the pulmonary capillary. And I'll just abbreviate that to make it easy. And what about the CO2? Which way does it move?
Goes from blood to air.
Or we could say from the pulmonary capillary to the alveolus. Oops. Excellent. Does that make sense?
Yeah.
Excellent. All right, so I'm going to cheat and make this smaller so that I can cram this up here as a reminder for us. Because once that gas exchange takes place, once oxygen is added to that blood in the pulmonary capillary, once CO2 is taken out, then obviously those gases have to be transported around the body. Now, technically, is this transportation around the body really a function of the respiratory system?
No.
What is it a function of?
Cardiovascular.
Yeah. All right. Of course, the reason these gases are transported around in the blood is so that these gases can take part in internal gas exchange. So let's start easy. Obviously, we are still dealing with the gases of carbon dioxide and oxygen. However, just like we did before with internal gas exchange, let's talk about what it is occurring between. And once again, we can think in terms of structures, or we can think in terms of either mediums or substances. And again, I will let you guys decide which you want to pick. What are the structures or what are the mediums or substances where internal gas exchange takes place between?
The capillary and the cells.
Excellent. I like that. Although we want to be a little bit more precise in our answer to that, but you're absolutely 100% correct. However, to be more precise, we could say systemic capillaries to distinguish them from the pulmonary ones we talked about. And while it is the goal is to get these gases either to or from the cells, those cells are contained within tissue, right? So really the systemic capillary is exchanging between the systemic capillaries and I would say the tissues of the body, which again, include the cells. So it's not wrong to say cells, but I think tissues might be a little bit more precise, especially when we think in terms of the mediums or substances. What is the substance that is contained within the systemic capillary?
Blood.
Yeah, blood. Excellent. And what is the substance or the medium that surrounds the cells inside of the tissue?
The interstitial fluid.
Perfect. All right. So again, it totally would have been acceptable to say cells before, but notice we say tissues, it matches a little nicer with the interstitial fluid. And then lastly, the direction for the two gases. Which way does oxygen move in this internal gas exchange?
From the capillary to the tissue.
Excellent. All right. Or we could think of this in terms of from the blood to the interstitial fluid. And which way does CO2 move?
Just the opposite.
Yeah, from the tissue to the systemic capillary, or from the interstitial fluid to the blood. Excellent. Does that make sense?
Yes.
Excellent. So again, let's make it, I don't want to erase it, but let's make it small so we can move it out of our way. Because what is the whole reason for us moving these gases out of the air into our blood or out of our blood into the air? is, of course, because of a process we call cellular respiration. And let's start easy with this. Where does cellular respiration take place? I agree.
Mitochondria, sorry.
Nope, they were enthusiastic about the answer. I appreciate that. Excellent, absolutely, in the mitochondria. Right, in the mitochondria, we use that oxygen to do what?
Break down glucose into water and CO2.
Excellent, to break down that glucose completely. And when we break that down completely, we generate a tremendous amount of energy, and that energy is used to make ATP. But you are correct, there are some waste products in that as well. Water is one of those waste products, and also carbon dioxide. So in that cell, at the mitochondria, we have this process of producing ATP so the cell can do all the work that it needs to do. And ultimately, this is the whole reason because you need oxygen to completely break down that glucose, and you're producing carbon dioxide as a waste product, which needs to be expelled from the body. Excellent. So notice when we talk about respiration, these are all of the processes involved. However, when we are talking about the respiratory system, which of these processes are really the ones that are a part of the respiratory system?
All but the gas transport?
Okay, I could see that, absolutely. Although you could argue that internal gas exchange is part of the cardiovascular system. Remember, we learned about capillary exchange there. And while cellular respiration has the name, if we were somehow able to get oxygen into the body some other way, then I think that we wouldn't necessarily call it part of respiration. So again, I love your idea. You absolutely have the right idea. Obviously, all of these things are related. But when we really talk about the functions of the respiratory system, really it's just these two. The movement of air into and out of the lungs, and then that external gas exchange between the air that we're moving and the blood in our pulmonary capillaries. All right. Excellent. And here's a pretty picture showing all of these processes there. Again, we kind of already did all of this, so I'm not going to go through it again. This was just, again, as a reminder, but we did all of this on the last page. Again, pulmonary respiration is just that mechanical act, or what we call ventilation. As we talked about, external gas exchange takes place between the alveoli and the pulmonary capillaries, where internal gas exchange takes place between the systemic capillaries and the tissues, all leading to that cellular respiration. So we did all of that. All right, so our function, to go back a page, for the next 3 lectures is going to be to focus on this. We will talk about these other things, but our focus is going to primarily be here. Now, like many organ systems, like many things in the body, we can identify them functionally and structurally. What is the function of the functions, plural, of the respiratory system?
Gas exchange.
Okay, excellent. That external gas exchange. What else?
Air filtration.
Excellent. All right, so that external gas exchange, we also talked about that ventilation, the movement of air. However, And I think we talked about this in terms of the digestive system. Remember, in the digestive system, we said we could put a straw up our nose and we could actually get milk into our stomach through our nose. Does that make our nose a part of the digestive system? No, of course not. Excellent. By the same token, anyone who has ever had a cold or the flu or really bad allergies, the only reason they survived that massively congested nose is because they were able to get air into their lungs via their mouth. We have that ability to move air into and out of our mouth. However, our mouth is not considered part of the respiratory system. And this air filtration is the primary reason. As we will see, our nasal cavity is not just specialized to move air to the lungs, but even though I have this chunk of Sacramento air right here in my hands, as you know, in Northern California, there's all sorts of horrible stuff within this air. You wouldn't want to take this and stick it straight into the lungs, right? There are smoke particulates from all of the fires going on, and allergens and dust and debris and all those other things in that chunk of air. So we need to do three things to it. We need to warm it, we need to moisten it, and we need to clean it. And our nasal cavity is specialized to do all those things, whereas our oral cavity is not. And so this additional function of filtering the air on its way is what makes the oral cavity not a part of the respiratory system. Again, like I said, vital from a functional standpoint, but not considered part of the respiratory system. Now, along those respiratory functions, we could probably throw smell into the respiratory function the same way we would put taste into the oral, but it also plays a couple other really important roles in our body's function as well. Respiration plays a huge role in the regulation of the pH of our blood. And my question for you is why? What is it about the respiratory system that helps us to regulate pH in our body? Does anybody know? There's this mystical, magical chemical reaction that takes place inside of our body. We'll actually talk about it in much more detail later. But for right now, it turns out when carbon dioxide is mixed with water, what do you get as a result of that?
Bicarbonate.
Right, exactly. True, you will get bicarbonate and hydrogen ions, which are from dissociated what? You're really gonna make me write out the whole equation? Okay, I can do that. Oops, CO2 plus H2O gives us H2CO3. Anyone know what H2CO3 is? Well, it gives us, as you mentioned, it gives us that bicarbonate ion, but it also gives us a hydrogen ion. You could even say that it is a hydrogen ion donator. And if it's a hydrogen ion donator, what does that make it? Again, don't get me wrong. It warms the cockles of my heart when you guys don't know chemistry stuff, because chemistry is stupid and nothing good has ever come from it. But H2CO3 is what?
An acid.
Yes, carbonic acid. It's an acid. So when you put water and carbon dioxide together, you get an acid. So as the levels of your CO2 in your blood changes, the pH of your blood changes. And is it important to maintain an appropriate pH level of your blood?
Yes.
Yeah, absolutely. I'm glad that from the very beginning, we've been talking about oxygen and CO2, because too often when people think about the respiratory system, they only think of oxygen. In many instances, well, don't get me wrong, oxygen is vitally important, right? You would die without it. But in many of the respiratory functions that you have, carbon dioxide is actually more important than oxygen, right? What do you do when you don't get your way?
Scream.
Okay, that's one thing that you can do. Or what else? You could do the opposite of that as well. What would that be?
Cry.
True. Or like what I do, I don't want to go to school. And you hold your breath, right? And your wife says, tough, you have to teach today, get out of here, right? But when you're holding your breath, can you hold your breath forever? There you go, Ariel's got it. No, absolutely not. Eventually you're going to breathe, right? You're not going to die from holding your breath. Eventually you will breathe. Do you breathe because you run out of oxygen?
No.
No, you breathe actually because you build up too much CO2 in your blood. Or at least that's the theory. Part of your home respiratory lab, we'll see if that's actually true or not. But for now, we'll believe that carbon dioxide is actually more important for regulating your rate of breathing than oxygen is. And then you can test that to see if it's actually true or not. Absolutely. So this carbon dioxide in our blood affects the pH of our blood. So it is vitally, vitally important that way. Of course, our nasal cavity plays an important role in our production of sound. Back in ancient times, people used to talk to each other over phones instead of just texting them. And when you called your best friend on the phone and they said hello, you could instantly tell that they were sick. How could you instantly tell that they were sick?
Sounded congested.
Yeah, the sound of their voice. With that congested nasal cavity, the sound, the tone of the voice changes dramatically. So the nasal cavity and paranasal cavities play an important role in our ability to phonate, produce sounds. So it plays an important role in that as well. All right. So all of these are functions of the respiratory system. As I mentioned, the body can be divided functionally and anatomically in many different ways. Your book does make a point of emphasizing both of these, but I'll be honest, I think the anatomical division of the respiratory system is stupid. But if we were to do that, basically what you do is you take a nice big fat black line and you run it right through basically the Adam's apple. And as you run it through the Adam's apple, you get the upper and the lower respiratory system. There are some doctors that specialize just in upper respiratory or lower respiratory issues. And I think that's basically whether or not someone wants to deal with the head or not. It's, again, I won't even say the word important. It's a distinction for whatever it is, but I'm not sure it's necessarily relevant. I think it is much more significant to divide the respiratory system functionally. Now, as we talked about, there were many functions, but there were those two primary ones. The two primary ones are gas exchange and getting the air to the lungs for gas exchange. And remember, part of that is the processing. And so that's what we have. Our conducting zone is the part that is responsible for the movement of the air and for the processing of the air. Right? Again, we want to clean it, we want to warm it, we want to filter it. So the conducting zone is everything that is responsible for that, whereas the respiratory zone is actually where the external gas exchange takes place. This is where oxygen and CO2 either enter the body or exit the body, right? This is where we cross the membranes. For that gas exchange to take place from outside to inside. And I think that is a much more significant division for the respiratory system, to think of it in terms of function. Yeah, there's nothing wrong with the anatomical distinctions. It's just, it's not like all of the upper does one thing and all of the lower does something else. Then that's not a case at all.
Professor?
Yes.
The warming of the air, does that help with gas exchange or?
I have no idea. No, I'm just kidding. Yes, absolutely. As we know, the warmer particles are, and this is true for our particles, but especially in gases, the warmer those gases are, the faster the molecules move. And actually, this is probably a great point. I'm glad you brought this up. This is actually a great point to bring up. As we just finished talking about moving oxygen and carbon dioxide into and out of our, across our external gas exchange membrane is a vitally important process. How long can you survive without oxygen? Now, if someone holds a bag over your head, or a plastic bag over your head, you're gonna last days, weeks,
Maybe a
Probably
couple
minutes.
hours? Yeah, a couple minutes, right? How long can you hold your breath for until you could pass out as a result of that?
minutes.
Yeah,
maybe?
again, we're talking a couple minutes. So getting these gases moved into and out of our body are hugely, hugely important. And yet, as vastly important as this process is, it is completely, assuming I could spell completely correctly, passive. And what do I mean by passive in this case?
No ATP.
Yeah. Does not use ATP. And since it doesn't use ATP, we have to do everything we can to facilitate this process and make it as efficient as possible. So our respiratory membrane is going to be tiny, so it has a very short distance to travel. Our respiratory membrane is going to be massive. Inside your chest, you have the surface area of close to a small tennis court. So that huge surface area for gas exchange to take place. And as you mentioned, by warming the air, we can get these gases to move more rapidly. And that helps to make the diffusion occur more quickly. It's also why we moisten the air as well. The gases actually move faster when they're dissolved in fluid. And again, you may never have thought about the fact that your respiratory passageway cleans and moistens and warms the air, but you're aware of it, or at least the past couple of mornings, if you've got to get up early to take your kid to high school like I do, when you step outside and you breathe out, what happens?
You see your breath.
You see your breath. Why do you see your breath? Because as that warm air comes out into the cold, with all that moisture in it, it condenses. And when it condenses, you can actually see the water vapor. So we've all seen our breath before, and the reason you're able to do that in the cold is because we are warming it and we are moistening it. Because gas exchange is passive, does not use ATP. As vital as it is, it does not use ATP. So thank you for bringing that point up. That's a great point to make here. We'll make it a lot, but this is a good starting point to make that. All right. Any other questions?
I had a weird question.
Yeah.
Is there any time when you're not warming the air in your system at all? Or is it just kind of always going to be warming the air?
I think, well, okay, so here's what I would say. The way we warm our air is by having blood vessels in our nasal cavity that are close to the surface of the mucous membrane. So they radiate off heat. And as you know, our blood is a couple degrees warmer than our core body temperature. So they're radiating off heat at a 99 degrees Fahrenheit or something close to that. So I guess technically, if you were like in a sauna, if you were in an environment where the air was warmer than your blood would be, then technically the air coming in would actually be slightly cooled by the because again, it brings it to whatever its temperature is. So it would still be warmer than it would still be warm, Yeah, I guess if you were in a really like in a desert, or if you're in a sauna or something like that, where the temperature was, 101, 102, then it wouldn't need to warm the gas in that point. Does that make sense?
Yeah, thank
Yeah.
you. There's just
Yeah.
a random thought that popped in.
Yeah, no, it's a great question. I hadn't thought of it before, but yeah, basically the way we warm it is by using our blood. So if you were inhaling air that was warmer than the temperature of your blood, then you wouldn't be able to warm it, but B, you wouldn't need to warm it. Does that make sense?
Yes, thank you.
Excellent. All right, spectacular. Excellent. All right, so let's talk about the anatomy. And as you can see, some of this anatomy is stuff we are already familiar with, things that we talked about in the skeletal system, or even things we talked about in the lymphatic or the digestive system. Air enters the nasal cavity via the external nares. Those are the two holes on the underside of your nose. They feed into this not elastic wouldn't be a right time, but rubbery pouch-like region, right? This is where you stick your finger when you pick your nose, and that is your vestibule. This vestibule, aside from being able to accept your finger, is lined with hair-like structures that help to limit insects or large dust and debris from coming in. And what would we call those hairs? We're not going to get away with a term like nose hairs. So what would the appropriate term for that be?
Cilia.
Cilia is not a bad guess, but remember, cilia are at the cellular level. So you are correct. The mucous membrane is going to be lined with a cilia, and we'll talk about that just a minute. But the large hair-like structures that if you look in the mirror, you can see sticking out of your nose, or that you trim with your scissors or your, manscaping or whatever that you use for that. What do we call those nose hairs?
Is it the nares?
Well, the external nares is the opening. In the vestibule, you have the vibrissae. So vibrissae is the name of those hair-like structures that, again, keep the large dust and debris and insects and things like that out of your nasal cavity. If we think in terms of bones, what bones are responsible for defining the space that is the nasal cavity? Let's rattle them off.
Maxillary?
All right, excellent. Oops. What else? Lacrimal. Yeah. I'll give you that. The lacrimal is really more. Here's what I would say with the lacrimal. Let's put an asterisk by that one. The lacrimal is really more of the orbit of the eye. However, it does help to form a passageway that for an opening for the lacrimal duct that does basically here. Let me see. Hold on. What's my next picture? No. So let's do this instead. Never find the pictures when I want them. Oops. Here we go. All right, so the lacrimal gland is part of the orbit of the eye, but what does happen is that from the lacrimal that is over in the orbit of the eye, it does help to form a passageway where the lacrimal duct will come down, and it actually enters the nasal cavity just lateral of this large bump. So if we were to cut a hole in this large bump, through that hole, we would actually see that opening on the other side. So I wouldn't mark it wrong on the exam if you said that because of that lacrimal duct. Let's see, we had maxilla, but let's at least put a asterisk by it. All right. What else? What bone did I just show where the lacrimal opening is located? What bone is that? This bone right here. What's that bone?
A vomer.
That is not the vomer, but you are correct in that the vomer is one of the bones that would help to form that intermediate septum, the midline septum of the nasal cavity. What else? Give me any bone.
Ethmoid.
Ethmoid. Excellent. Ethmoid helps to form the top up here, and its perpendicular plate is also with the vomer going to form that nasal septum. What else?
Palatine.
Palatine bone. Excellent. With the maxilla, it forms the floor of the nasal cavity, what we think of as the hard palate we just finished talking about in the digestive system. What else?
Nasal bone.
Nasal bone, excellent. We can actually see the nasal bone, which is one of the nice reasons why I switched to this picture. We can see the little bit of the nasal bone helping to form part of the bridge of the nose. Excellent. Notice there's a bone here between the nasal bone and the ethmoid bone. What would that be? With this nice big space inside of it.
The frontal sinus.
Frontal sinus of the frontal bone, exactly. I see another sinus here as well. What contains, what bone is that sinus a part of? Right here we have this sinus. Sphenoid, there you go, Amy's got it. Excellent. Of course, the ethmoid not only forms the roof of it with that cribriform plate, but also helps to form these two bumps, the superior and the middle nasal concha. But that still brings us back to this bone. What bone haven't we identified yet? Come on, if the two bumps above them are the superior nasal concha and the middle nasal concha of the ethmoid, what is the bone that is inferior to those two?
The inferior concha.
Yeah, the inferior nasal concha, which remember is its own facial bone. Excellent. I think that's a pretty good list. I think that does a good job of defining it. Notice those definitely form the floor, the walls, the ceiling, and with that perpendicular plate of the ethmoid and the vomer, we also get that midnasal septum, right? If we, I think if we jump back a couple pictures here, let's start here. I don't want to erase the words. Oops. Here, this one happens to be slightly deviated, but we can see the perpendicular plate of the ethmoid and the vomer forming that nasal septum. So unless you've been snorting massive amounts of cocaine, you cannot put a handkerchief up one nostril and pull it out the other. Unless I guess you get a piercing that's big enough, then I guess you could do that too as well. So I guess I have to, so either you're a coke fiend or you have a piercing, you can't get it from one to another. One of the interesting things about this nasal septum is it does indeed divide the nasal cavity into two separate compartments. This goes back to what we were talking about before with the filtering and the processing of the air. Within the walls of your nasal concha is actually erectile tissue similar to the erectile tissue in the penis and in the clitoris and vagina, or labia actually. And The reason for this is that if one side of the nasal cavity is a little more dry or is a little too cool, where it is not able to optimally filter the air, then that erectile tissue will cause that side to become congested, encouraging the air to go through the other side. And it will alternate back and forth between those to, again, maintain optimal conditions for filtering the air to process that air properly. Excellent. All right. Since we're here, I'll go ahead and show this as well. The air is going to leave the nasal cavity from the internal nares. The internal nares is basically this opening right here, formed by the floor and the walls of the palatine bone, the perpendicular plate of the ethmoid, and the minor wings of the sphenoid, the greater wings of the sphenoid are all going to help to form this opening. I've got a couple other nice pictures. There's another one. Here we see our nasal septum. Again, here is the vomer. Here is the perpendicular plate of the ethmoid. And notice if we were to peel that aside, we would see those three bumps, the nasal concha. Again, remember the superior and middle are bone features of the ethmoid, but the inferior nasal concha is a bone of its own. Notice also there are these passageways inferior to the concha. These are the superior, middle, and inferior meatuses. What's the point of having all of these bumps? And then also our frontal sinus, our sphenoid sinus, our ethmoid sinuses, and way laterally over there to the side, we have the maxillary sinus for paranasal sinuses. And I've got a pretty picture that'll show that to you in just a minute. Why are they important? Why is it significant? Why not just have it a straight hollow tube like the oral cavity is?
Is it more surface area for filtration?
And not just more surface area, but again, if the air just races by, are you going to have time to clean and warm and moisten the air?
No.
No. We want to swirl the air around. We want to produce turbulence. Having these concha, having these paranasal sinuses produces turbulence, which swirls the air around in this space, so it doesn't immediately go into your lungs. And as it's being swirled around, that swirling basically does two important things. First, it brings the chemicals up to the superior portion where all our olfactory nerves are, so we can smell whatever is in the air. But by swirling it around inside of here, it gives us more time to process it before it leaves to go to the lungs. There we go. Oh, perfect. Love this picture. Notice here again, we see that internal nares, the bony structure that forms the internal openings of the nasal cavity. And notice if you look closely, we can see the inferior, because remember this is an underside view, inferior, middle, and tucked right down in there, we can see a little bit of it, the superior nasal concha, producing all of that swirling around for the air in those spaces. And we can see something similar. Here we see an actual intact cadaver. Notice in this intact cadaver, we can also see the vestibule, again, that pouch-like region, and the vibrissae that are filling it within there. And now that I've drawn them, I'll erase it so we can see that. Notice we can see the inferior, middle, and superior nasal conchas, and then of course the meatuses traveling beneath them. And then we can actually see where the bones form that internal nares. Notice the soft palate extends beyond as well, but there's no bones in that. And as we know, that's going to help anything in the oral cavity from getting up into that nasal cavity. All right. So we did that, we did that, we did that. We mentioned this briefly, but we didn't talk about it in detail. What type of membrane lines our nasal cavity?
Mucous membrane.
Excellent. And like all membranes, it is an epithelial tissue sitting on top of a connective tissue. What is the epithelial tissue forming the mucous membrane in the nasal cavity?
Ciliated.
Pseudostratified epithelial tissue.
So a ciliated, pseudostratified columnar epithelium. And what is the connective tissue?
Areolar.
And what do we call it based on its location?
Lamina propria.
There we go. Perfect. The job of those cilia is, as we mentioned, it's the mucous membrane. It produces mucus. That mucus helps to warm the air that passes over it, and it helps to capture the dust and debris. And then the job of that cilia is to move that mucus to the nasopharynx, where it can then either be swallowed, where any dust, debris, and pathogens can be destroyed by the hydrochloric acid of our stomach, or it can be spit out, getting it out of our bodies. Excellent. And we talked about the septum. We did all of that. We did the mouth as well, so we don't have to do that as well. Excellent. Here's the picture that I promised of those 4 paranasal sinuses. We have the 2 lateral maxillary sinuses, the 2 frontal sinuses, right? A singular sphenoid sinus right on the midline. And then within the walls of the superior and middle nasal concha of the ethmoid are a bunch of air-filled spaces. In fact, if we cheat, if you remember way back in 430, whoops, where'd it go? When you look at the ethmoid has all these air-filled spaces within it. And then, as we also talked about up here at the top, we have the cribriform plates, the flat part. And what sits on the cribriform plate?
Crista galli.
Well, the crista galli is the big fan thing that sits between them. So it does sit between them. But what actually sits on the crista galli? What is the structure that sits on the crista galli? Maybe if we go back to this picture, it'll help. What is this elongated structure right there that sits on top of the cribriform plate?
Is it the olfactory?
Yeah, it's the olfactory bulb. Off of that olfactory bulb is the olfactory nerves that penetrate through the olfactory foramina into the superior part of the nasal cavity, providing us with our sense of smell. Those of you who had me for 430 have heard this story before, but my sister was dating a young gentleman who got incredibly drunk one New Year's Eve and fell down a flight of stairs. And as he was tumbling down those flight of stairs and his brain was rattling around inside of his skull, his olfactory bulb ripped off of his cribriform plate. And so when he sobered up and woke up the next morning, guess what he could no longer do?
Smell.
Yeah, he lost his sense of smell, right? And still has no sense of smell to this day, some 20 years later, right? But the story does have a happy ending. She did break up with him and married his best friend. So it's all turned out great in the end. All right, excellent. No, what do I want to do now? Back to. Yeah, excellent. I love some of these pictures from your textbook. Notice this is the posterior side of a coronal section. There you go. Never forget the function of the olfactory bulb. I love this picture. I think it's so, so cool. And because it shows so many of these great features that we are talking about here. Again, notice here we see where the vomer and the perpendicular plate of the ethmoid are dividing the nasal cavity in half. We can see where the middle, inferior, middle, and a little bit of the superior nasal concha are there. And then the meatuses around them. We can see that lateral maxillary sinus, that frontal sinus up here at the top. So again, this is a coronal section that we are looking at the posterior view of. And I think it does an awesome job of showing many of these specializations. And as we talked about, the purpose of all of these specializations is to produce turbulence, to not allow the air to immediately go into our lungs, to give more time to process it, to slow it down, swirl it around, so we get our sense of smell, so we get our cleaning, our filtering, our moistening, and our warming of the air. All right, questions on that? All right, as I mentioned, it exits the internal nares, which is that bony feature that we saw here, formed again by the L-shaped palatine bone, the ethmoid bone, and then the sphenoid bone, forming those openings. So we can see that nicely here. And then notice it leads from there into our pharynx, which we talked about last time. Notice our pharynx, as we talked about last time, is divided into three parts: nasopharynx across posterior to the nasal cavity, oropharynx across from the oral cavity, laryngopharynx opposite of the larynx. Remember, we talked about the pharyngeal tonsil located at the top of the nasopharynx, the palatine tonsils in the lateral walls, basically between that palatoglossal and palatopharyngeal arches. Our lingual tonsils we can see here. We can even see that opening to the auditory tube, which remember we talked about in 430, connects to the middle ear, where our auditory ossicles vibrate to produce our perception of sound. While we're here, while we're thinking about it, we talked about the epithelial tissues of these. The nasopharynx, what is the epithelial tissue that lines it? Come on, we just did this in the last section. What is the epithelial tissue that lines the nasopharynx?
Ciliated columnar.
Yeah, it's the same as in the nasal cavity, ciliated pseudostratified columnar epithelial tissue. Because remember, the nasopharynx should only contain air. Conversely, does the oropharynx and laryngopharynx just contain air? No, they're passageways for air, but also for food and drink. And as such, both the oropharynx and laryngopharynx are lined with what type of epithelial tissue?
Non-keratinized.
Exactly, a non-keratinized stratified squamous epithelial tissue. Exactly. Excellent, excellent. So again, we did that, we did all of that. And we talked about this stuff as well. So we've identified that, again, oropharynx has the non-keratinized stratified squamous epithelial tissue, whereas our nasal cavity and the nasopharynx have that ciliated pseudostratified. We know that all of this is a mucous membrane, so it is an epithelial, an areolar connective tissue that, based on its location, we call a lamina propria. Not surprisingly, most of the upper respiratory tract, it contains a large amount of mucus glands for producing that mucus. As we mentioned, our nasal cavity has a lot of superficial blood vessels. those blood vessels radiate off the heat that helps to be able to warm up the blood. What's the downside of having all these superficial blood vessels in the nasal cavity?
If you get punched in the nose, so bleed a lot.
Yeah, punch the nose, get a little bit too aggressive, picking a booger, right? Or it can just simply be irritation from allergies or from dry air or from being around the smoke. That even just minor irritations like that can actually cause those superficial blood vessels to bleed, causing a bloody nose. And we already talked about the olfactory receptors and my sister's ex-boyfriend, so we don't need to worry about any of those things. All right. Lastly, as we mentioned, we just talked about the pharynx with those three regions. And again, as we talked about, the nasopharynx is air only, whereas the oropharynx and laryngopharynx is air plus food and drink. And the biggest difference we see between these because of that is what type of epithelial tissue lines them. Again, our nasopharynx is, again, that ciliated, pseudostratified, columnar epithelial tissue, whereas the oropharynx and laryngopharynx is that non-keratinized stratified squamous epithelial tissue. Oh, that didn't write. There we go. Excellent. All right. Questions on that? All right. From there, we enter into the larynx. As we talked about in the last section, we know that the epiglottis of the larynx and the elevation of the larynx is going to be an important part of our deglutition process, or the process of swallowing. But so obviously it plays a very important role in making sure that air goes into the trachea and food and drink goes into the esophagus. But it also plays a couple of other important roles and has a fairly elaborate anatomy. And so even though it is a teeny bit early, I think this is a good natural stopping point. So let's go ahead and take our first break here. It is, let's get these process 102. So let's restart at 1:17, and I will start the recording at that time. All right, so any questions on the pharynx? Again, shouldn't be too difficult because we just finished talking about it in the last section, but any questions on the pharynx before we finish that up and take our first break? All righty then I will see you in 15 minutes. All righty. So let us talk now about the larynx and its anatomy and its function. Now, as we just finished talking about, we know the nasal cavity and the nasal pharynx is lined with a ciliated pseudostratified columnar epithelial tissue. And we know that the oropharynx and the laryngopharynx and the proximal part of the esophagus, for that matter, are all lined with a non-keratinized stratified squamous epithelial tissue. So what type of epithelial tissue do you think we're going to find in the trachea and in the larynx? I did turn on the microphone, right?
Yeah.
Okay, excellent. So what do you guys think? Well, let's see if we can tease it out. What passes through the nasal cavity again?
Air.
Air. What passes through the oropharynx and laryngopharynx?
Air and food.
Air and food. What passes through the, what passes through the larynx and the trachea? Just air. And so if it's just air again, what do you think it's going to be lined with?
Ciliated.
Yeah, it's again going to be a ciliated pseudostratified columnar epithelial tissue. In fact, here it is what is often referred to as the mucus elevator. You may remember we talked about this in 430. As we mentioned, these cilia help to move mucus, but they're delicate. So if you are out in the cold, for instance, for a long period of time, your nose starts to run. However, these cilia are also irritated by smoke. So if you are a chronic smoker, that smoke can irritate the cilia, so they become non-functional. So now when that smoker produces that mucus in their larynx and in their trachea, how do they have to move it?
By coughing?
Yeah, they got that great smoker's hack to be able to get that out of the airway. It's one of the reasons why if you're a chronic smoker and you get a cold, you have to be really, really careful about taking cough suppressants. Because if you take cough suppressants, that depresses your cough reflex. Your poor little cilia can't move that mucus, and so all that mucus and that dust and that debris instead drains down into the lungs, dramatically increasing the likelihood of pneumonia or even lung cancer, other types of respiratory issues, COPD, things along those lines as well. Excellent. So once again, we're in an air-only pathway. So not surprisingly, it is lined with a ciliated pseudostratified columnar epithelial tissue. There are two folds in the lining, and we see this nicely in the illustration if we get rid of everything else. There is not one, but two folds within the line of the and within the mucous membrane of our larynx, and they form what we commonly refer to as the true and the false vocal cords, but more appropriately are referred to as the vestibular fold and the vocal fold, and we'll take a closer look at those in just a minute. So we have those. Oops, sorry, go back. Excellent. Here, with all the skin and muscle peeled away, we see what the larynx looks like. Its job is to, again, not only to allow the food and drink to go into the esophagus, but to allow the air into our respiratory passageway. Unlike the esophagus, which we know is just a muscular tube that pretty much collapses when it's not in use, our airways aren't something we want to collapse. And so instead, it is held open rigidly by cartilage. In fact, there are 8 rigid pieces of hyaline cartilage that help to hold and stabilize this airway and keep it open. And then, of course, there is that one elastic cartilage structure. Remember our epiglottis that we talked about, who's responsible for bending and folding over the top when the larynx elevates to close off that passageway. Now, if we go back to the previous picture, as you can see, the largest of the cartilage pieces is this big, huge shield-like structure located in the front called the thyroid cartilage. And why do you think it's called the thyroid cartilage?
Located around the thyroid?
Close, not quite around, but superior. The thyroid basically, well, okay, that's a horrible drawing of the thyroid. That we got the 2 lobes and the isthmus of the thyroid, which basically sits just inferior to the thyroid cartilage. So it sits underneath it, but you're right, it's right next to it. So it doesn't sit on top of it, but it does sit right next to it. However, also notice is there this big, large shield-like structure, but notice also there is a pronouncement that sticks out anteriorly, known as the laryngeal prominence, or what is commonly referred to as the Adam's apple. Of course, it's called the Adam's apple, but are males the only ones that have this laryngeal prominence?
No.
No, everybody has them. However, upon puberty, the androgens that are produced by males do cause, among other things, our cartilage to thicken and enlarge. And so the laryngeal prominence is typically more pronounced in males. And so that more pronounced is because of those androgens. Actually, many women find it a attractive characteristic. And if you think of it from a biological standpoint, that kind of makes sense, because a male that is producing a large amount of androgens is one that, from a biological standpoint, might be viable as a mate. So again, so that's often something that is found as a desirous characteristic. However, while this thyroid cartilage is the largest chunk of cartilage, as we can see here from the illustration, it doesn't actually go around the entire larynx. It forms instead a C-like structure. And in fact, there is only one piece of the 8 pieces of hyaline cartilage that form the larynx. Only one of those pieces is a completely enclosed circle. That piece is inferior to the thyroid cartilage, known as the cricoid cartilage. Notice the cricoid cartilage is much smaller on the anterior side, much wider on the posterior side, and we see that nicely when we look at it in this sagittal section. We can see that it has a much larger base posteriorly and is much more narrow anteriorly. So we see that. And again, that thyroid cartilage sits on top of it, or around it, I should say, superior to it. What does sit on top of it are 2 additional pieces of cartilage. Notice from the posterior and lateral view, there is this, oh, I already used green. There are these triangular pieces of hyaline cartilage that are sitting on top of that posterior base of the cricoid cartilage. Those are known as the arytenoid cartilages, and they are paired. There is 2 of them. Notice there's only 1 thyroid and only 1 cricoid cartilage. And then if you look closely, you will see that superior to the arytenoid cartilage are 2 little corn kernel looking pieces of cartilage. And that's how I remember them. They look like a little kernel of corn, and they are the corniculate cartilages. And those are also paired. There are 2 of those. And so as you can see, with that, we have our 8 pieces of hyaline cartilage that forms our larynx. Questions on that? Really? No one has a question on that? I can wait if you need to take off your shoes and socks to do the math.
You only counted 6.
There you go. Excellent. Absolutely. There are indeed only 6 that I have identified. And that is because there is one other paired piece of cartilage that we don't see in this illustration. Notice this illustration does a nice job of showing the cartilage, showing the ligaments that are holding this cartilage together. Notice it even shows us our hyoid bone, that U-shaped bone that was so special. What's so special about the hyoid bone again? Anyone remember?
Was it the only free floating bone?
Absolutely. It is the only bone that does not articulate to another bone. Be careful, it does attach to the body. It is held in place by ligaments and tendons and muscles, but it is the only bone that doesn't form a joint. It does not articulate to another bone. Instead, it acts as that movable base of the tongue. All right, so we see a lot of stuff here, but we don't see everything. What we are lacking, what this picture doesn't show, in this posterior view is probably a nice way to see this. There is a fold in the mucous membrane that basically travels from the epiglottis and comes down basically to where the arytenoid cartilage is located. Any idea what that fold in the mucous membrane that goes from the epiglottis to the arytenoid cartilage might be? So if only I had the list in front of me of my study guide helping me with my anatomy that I'm responsible for on this exam, I might be able to figure out what the fold in the mucous membrane that goes from the arytenoid cartilage to the epiglottis was. I'll wait.
Is it area epiglottic?
Fold? There you go. See how easy that was? Absolutely. It is indeed the aryepiglottic fold. Notice with this slightly different picture, we see that aryepiglottic fold. We see that fold in the mucous membrane that goes from the epiglottis down to the arytenoid cartilage. And notice embedded within that membrane, not attached to any of the other cartilage pieces, is basically a little piece of cartilage that helps in the folding of the mucous membrane when the epiglottis comes down. And these two chunks of cartilage free floating within this mucous membrane are known as the cuneiform cartilages. Notice our illustration shows this really nicely and it highlights it inside of the membrane. But there are a couple other ways we can see this as well. Let's switch to this view. I have some pretty pictures here. No, hold on. There you go. You guys seeing that?
Yeah.
Excellent. Hold on. I had to close my chat window. Let me bring it back up again. Perfect. So what you can see here, this is a posterior view of the larynx. We can kind of tease some of that out because we can see the hyoid bone going around to the back there. We can see the broad base of the cricoid cartilage. However, the two dead giveaways that are going to help us to see what we're looking at is here we have the epiglottis, that is that elastic piece that's going to fold over. Obviously, it is connected to the aryepiglottic fold. And if you notice in that aryepiglottic fold, there is 1, 2, 3, 4 bumps. The two bumps that are inferior and more medial are the corniculate cartilages, and the two bumps that are superior and lateral are the cuneiform cartilages. So we see that nicely on a simple model like that. Notice again here, this simple model still shows us those two bumps here on the back that we're going to be able to distinguish it. There's a couple nicer ones here. Notice with this one's not a good picture of this particular model, but as you can see, the corniculate cartilage helps to form these two bumps here. And if you look closely, like I said, this is not a good view of this particular model. There is two bumps there. That is the cuneiform where we can see stabilized in there. This, of course, is our aryepiglottic fold to the epiglottis. Our arytenoid cartilage, corniculate, sitting on top of the cuneiform. I think I have, no, not what I wanted. The other place we can see this really nicely, and I haven't peeked to see if it's there, but I'm going to guess that it's there. Semnis River Colleges, Virtual Anatomy Lab, Respiratory System, charts. Oh, no, that's reproductive. So why is there we go. Charts. There we go. Perfect. Notice this is a picture we have seen before. We looked at it before when we were looking at the thyroid and parathyroid glands. But now notice we can see very clearly here our respiratory structures. Notice here we see the epiglottis. We can see the epiglottic fold that is coming around. And notice very, very nicely, we can see the 1, 2, and then I'll just put dots on them on this side, 3, 4 bumps that are formed by the cartilage on the posterior side of the larynx. And again, the 2 inferior and medial ones are formed by that corniculate cartilage sitting in on top of the arytenoid. And then within the aryepiglottic fold is those free-floating cuneiform cartilages. So there you go. The big thyroid one in front, cricoid on the bottom, 2 arytenoid, 2 corniculate, and 2 cuneiform. And there we have our 8 hyaline cartilages. And then our 9th is that elastic cartilage that is the epiglottis. Notice there's one other place where we can see this. Wow, this is the one I want. Oops, here we go. So again, we saw those, saw that. Is this here? Notice again in this superior view, if you were to take a camera down your throat to the larynx, this is what you would see, right? You would see, and this is what you're actually looking for when you're intubating someone. You're using that little tongue depressor thing to basically not just hold the tongue back, but also to grab a hold of the epiglottis so that you can slide the tube right here in between the 1, 2 folds of our larynx into our airway. And so notice here on the posterior wall, again, we have the 2 cuneiform cartilage bumps that are lateral. They're also superior, although this view doesn't show that well. And then the 2 corniculate, which are inferior and also more medial. So what happens, and let's go back to the previous picture now. The arytenoid and corniculate cartilages basically sit on top of the cuneiform, and they have the ability with these ligaments that we see to rotate out and in and out and in. And as they rotate out and in, they open and close the airway, what we call the glottis. Remember, we actually talked about this in the Valsalva maneuver, how when you close the glottis, we can force the air down to help in defecation instead of letting the air escape. Well, this is what we're talking about. This opening that is formed by those rotating arytenoid and corniculate cartilages is the glottis. And notice the door to the glottis is formed by not 1, but 2 folds in the mucous membrane. We have our vocal fold, which is inferior and medial. And we have our vestibular fold that is superior and lateral. But their locations are not the only thing that are different about them. Our vestibular fold is, as you can see, well vascularized. It's a part of the fold of the mucous membrane that is highly sensitive. So if any type of dust and debris gets into this region, it would irritate this vestibular fold and trigger a coughing reflex, where we would force a large amount of air out of the lungs to try to expel whatever that dust and debris is. Inferior to that, we have the avascular and thinner and also more rigid vocal folds. And those vocal folds are what vibrate to produce the sound that we use to make speech. So again, vestibular is superior, lateral doesn't have that flexibility that we have in the vocal folds, whereas our vocal folds have that elasticity. And that elasticity allows it to vibrate. Much like when you pluck a string on a guitar, the vibration of it produces a sound. And that is exactly what our vocal folds do. And how do you tune a guitar?
Tighten it or loosen it.
Right. By increasing or decreasing the tension on the string, you can get it to vibrate faster or you can get it to vibrate slower. And so as you change the rate of vibration, you change the tone of the sound. When it vibrates faster, what happens to the tone?
It gets more sharp.
Yeah, well, and it goes higher as well, absolutely, right? Whereas if you loosen it, it gets a deeper sound. And that's exactly what happens with our vocal folds. This is something we are consciously capable of doing. Right now, am I actually thinking about which precise muscle I am controlling to be able to get the right tension to produce the right sound for when I want to talk deep or when I want to talk high? Right? No, I'm not. I don't know which muscles I am actually controlling, but it is something I am consciously aware and capable of doing. One of the reasons we know this for certain goes back to that puberty we were talking about. In males, not only does the increase in androgens cause an enlargement of the Adam's apple, but it also causes a thickening of the vocal folds. And when we thicken the vocal folds, it doesn't vibrate at the same rate that it did before. So now when you contract those muscles to produce a particular sound, do you necessarily get the sound you expected?
Nope.
No. And so what can happen to that poor male's voice as obviously he gets deeper because again, the thicker vibrates slower, but he doesn't instantly have control over that. So when a male hits puberty, what often happens to them?
Voice cracks.
Yeah, they get that cracking of the voice. That cracking of the voice is from that incomplete control of the vocal fold because of the changes in the anatomy of that vocal fold. Now, one last thing. I have my microphone here on the table to pick up the sound as I am speaking. Wouldn't it be more efficient to take that microphone and put it all the way down my throat, down here to the vocal folds, so that when they vibrated, you could hear my speech?
No.
No. Would you hear my speech if you did that?
No.
In fact, if you were to put a microphone down there right next to the vocal cords, what you would actually hear is just a buzzing noise, right? The vibration of the vocal folds produce a sound, a buzzing sound that we call phonation. Again, the bigger the vibrations, the louder the sound. The faster the vibrations, the higher the sound. Slower, the deeper or lower the sound. So we do have some control of the sound that way, but it really is just kind of a buzzing sound. What we then do with that buzzing sound is we then shape that sound with our nasal cavity, with our oral cavity and our teeth and our tongue and the muscles of our throat. And we shape that into speech. And that process of taking that buzzing sound and shaping it into speech, we call articulation. Right? And as I mentioned, this is not new information. This is something we know because, like I said, you can tell when someone has a cold and they're talking because their ability to articulate is different. The tone and sound of their voice sounds different when their nasal cavity is congested. Or you can just simply hold someone's tongue and ask them to talk. Are they going to sound the same if you're holding their tongue while they're trying to talk?
No.
No, of course not, right? So we need to shape that sound, and that is the process of articulation. All right, questions on that?
Yeah, with people with the stomas, is that technically what the machine does? It articulates for them?
Yes, essentially that is what it does. Yeah.
Okay.
Yeah.
Sorry, I keep
No,
getting a question popping in my head.
It is a great question. It's a little bit more sophisticated than that, but in the most general sense, sky is blue sense, yeah, essentially that's what it's doing. Basically, it is providing that ability to do that. Yeah, absolutely. All right, excellent. So as the air passes from the larynx into our trachea, or what we commonly refer to as the windpipe, it connects to our bronchi, and it connects, again, lined with that ciliated pseudostratified columnar epithelial tissue, forming that mucus elevator that we talked about. At its base, it terminates to form two bronchi. And of course, why does it form two bronchi?
There's two lungs.
Yeah, because there's two lungs. And in fact, we call those the two primary bronchi. At the distal end, where the trachea branches to form the two primary bronchi, at the base of it is another area of highly sensitive mucous membrane. Remember, we talked about the mucous membrane of the vestibular fold being very, sensitive. Well, the carina is also very sensitive as well. If a tiny bit of dust and debris, or if you accidentally laugh while you're drinking and a droplet of water were to come down and make contact with the carina, that dust or that water will cause an incredibly violent coughing fit to try to, again, expel that irritant and keep it from getting into the dust, getting into our lungs. So it is a very important protective reflex by this highly sensitive fold of the mucous membrane known as the carina. And as I mentioned, it splits to form two primary bronchi because we have two lungs. Now, this is what the trachea looks like from an anterior view. However, we do want to take a cross section through it. And when we look at the cross section, well, let's look at it here first. When we see the trachea, we see that much like the larynx, it is stabilized with rings of cartilage. All right, these are rings of hyaline cartilage that again keep the airways open. However, when we take that cross-section view through it, we see that those rings actually don't go all the way around. They are just a C-shaped ring. They are actually open to the back. And the nice thing about the illustration, and also the nice thing about the light microscopy view here, is we can kind of get a sense why. Because notice that posterior to the trachea is where the esophagus is located. As we know, this esophagus has got to be able to accommodate that big, huge bite of cheeseburger that you took, right? I know everyone in this class chews it the 75 times you're supposed to before you swallow it down, but some people may take a bite, chew twice, and then swallow. And that bite could be half the cheeseburger. Right? The advantage of the esophagus is being a muscular tube. It can expand to accommodate that. But while it was expanding, if there was a rigid ring of cartilage here, that rigid ring of cartilage could limit the ability of food to pass. And that food could get stuck. And if it got stuck, that could push on the cartilage, decreasing the size of the trachea and blocking the airway, which would absolutely positively be a bad thing. Now notice, and again, our illustration does a great job of showing this, both in the illustration, but also as we can see very clearly in the light microscopy, we do still want to be able to control the diameter of our airway to be able to increase it or decrease it. So you'll notice there is a thin line of smooth muscle located at the posterior part of the trachea, known as the trachealis muscle. And that trachealis muscle helps to be able to change the size, the diameter of the airways. And of course, being smooth muscle is controlled by hormones and our sympathetic and parasympathetic, right? Sympathetic, what would it cause this trachealis muscle to do? Contract or relax? Well, when is your sympathetic nervous system active?
Under stress?
Yeah, in your fight or flight response. So if you're in a fight or flight situation, do you need to get more air or less air into your lungs?
More?
So would you want to constrict your trachealis or relax your trachealis?
Relax.
Relax, there you go. Conversely, if you eat that poisonous mushroom that stimulates your parasympathetic nervous system, it causes this trachealis muscle to constrict, decreasing the size of the airway, and all these mucus glands in your mucous membrane of your trachea become active. So we start filling that space with mucus and the airway is constricted, and suddenly it's a whole lot harder to get oxygen into your lungs. So there you go. So even if you hadn't thought about it, you know how these things work. Excellent, we did that. As I mentioned, we have two primary bronchi because we have two lungs. However, it is not the primary bronchi that enter into the lungs. Our primary bronchi do not enter the lungs. Instead, before they enter the lungs, they branch. And when they branch, what do you think those primary bronchi form?
Secondary
There you
bronchi?
go. See how easy that is? They form secondary bronchi. Any idea how many secondary bronchi we might have?
5.
Any idea why?
5 lobes.
Exactly. And in fact, our secondary bronchi are known as the lobar bronchi because we have 5 lobes to our 2 lungs. And so we have 5 secondary bronchi, and it is these secondary bronchi that are going to enter into the lungs. Excellent. Notice there is some asymmetry both to the lungs and to the bronchi. Notice that in the primary bronchi, the right is typically wider, shorter, and more vertical, whereas the left is longer, more narrow, and more oblique in its angle. Functionally, that isn't too significant, but from a clinical standpoint, why might that be significant? Why might it be clinically significant to know the anatomy of the two primary bronchi?
The structure of heart to give space to the heart.
True, you're right. That is the reason for the asymmetry, absolutely. It's because we have to accommodate that pesky heart that's sitting offset in the center of our chest, not quite on the midline. But let's think of it this way, from a clinical standpoint, if someone were to accidentally inhale a pee, is that pee equally likely to lodge itself in the right and the left primary bronchi?
No,
So which side is it more likely to lodge itself in?
the left side.
Actually, the right. Because of the vertical nature of it, it is actually more likely to wedge itself into the right. So blockages that occur from food are much more common on the right side than they are on the left. I like what you're thinking. Because it's more narrow, it might be more likely to occur in the left. But the fact that it's more narrow and more oblique in its angle makes it less likely that something's going to roll in there, where it's much easier for something to fall into the more vertical and the wider right side. All right. Notice there's that magic word hilum. That is a term we've used before. Like the kidneys, our lungs have a convex surface and a concave surface. Like our kidney, like our lymph node. And like our lymph node, the concave surface we call the hilum. The entire concave surface of the lung, which I should say is medial, is known as the hilum. Whereas the entire convex outer surface, which of course is anterior, posterior, and lateral, is the part that is in contact with the ribs, and so not surprisingly is known as the costal surface. Now, much like we saw in our lymph node, things enter into and out of the hilum. So in our hilum, we have our blood vessels and our nerves and our secondary bronchi that all enter into. So again, blood vessels, nerves, secondary bronchi. All of these, and I have to make this smaller. All of these enter into the hilum in a specific region known as the root. And so the root is just that area where those structures enter. So the root is a smaller portion of the hilum. I make a point of emphasizing this because there's one more thing we know about the lungs, probably more than one thing, but there's one thing that I know for a fact we definitely learned way back in 430 about the lungs. What did we learn about the lungs way back in 430? What's something we learned about the lungs back in 430?
Was that they're surrounded by a membrane, a serous membrane?
Absolutely, that they are surrounded by a serous membrane. And what would we call the serous membrane that surrounds the lung?
The visceral pleura?
Absolutely. So remember, we have a visceral pleura that is in direct contact with the surface of the lungs. There we go. Got there eventually. And what do we know about pleural membranes? Do they just come in the visceral the visceral variety?
There's always a parietal.
There's always a, oops, hold on. Let's keep that brown. We know there is going to also be a parietal pleura, and I'll cheat and draw it down here. That's going to line the thoracic cavity. And what do we know about visceral and parietal pleuras? Are they, are they completely separate and independent of each other?
No, they have like serosal fluid in the middle.
True, and they're connected. They're continuous with each other. The way this works is that the parietal pleura actually comes to those blood vessels and those nerves and those bronchi, and then that's where they connect to the visceral pleura. So notice the root of the, I didn't write root anymore, did I? The root, I'll do it over here. As I said, is the attachment point. I wrote this somewhere. I remember doing the secondary bronchi like that. Not seeing it, so I guess it doesn't matter.
Upright.
Oh, there you go. Excellent. Oh, okay, I did do that. Okay, I knew I did it somewhere. All right, excellent. Enter that. And now we can add to that definition. It is defined by the pleura. One of the nice places where you can see this is in the lung models that we have in the classroom. I'm not sure where I have a picture of these. Let me see what I've got. Don't have it there. I don't have a good model that shows this. Oh, here, this will work. We'll do that. Okay. So let's take a look at the lung and the lung anatomy, and then we can be able to see this. The lungs are cone-shaped structures. And like all cone-shaped structures, it has an apex and it has a base. Where is the base located of the lungs?
On top of diaphragm.
Yeah, it sits on top of the diaphragm. And any idea where the apex is? Basically,
At the top?
yeah, it comes up, and more specifically, it comes up to about your clavicle. So the apex is located about the level of your clavicle, and the base is sitting on top of the diaphragm. As I mentioned, the three outer surfaces is the costal surface. The entire, this is the picture I wanted to show you. The entire concave surface is the hilum, right? However, notice specifically within that hilum, there is this region where we can actually see the visceral pleura coming off of the walls of the lung to form the parietal pleura, and contained within that pleura as it comes off is the blood vessels, the nerves, and the airways. I make a point of emphasizing this because notice your book has its label for hilum, and it points here right smack dab in the center. Is that correct?
Is that more specifically the root?
You're correct, absolutely. It is correct, it's the hilum, because this entire concave surface is the hilum. However, you are also correct, and it would be much more appropriate for this label to refer to this specifically as the root, right? But the root is within the hilum, so they're not wrong, but that it's more specific. If I have that label there, I'm going to want you to be specific, and I'm going to want you to say root. All right, so we see that root very nicely. Here we see the costal surfaces of the lungs. And as was mentioned, we see that there are 5 lung lobes to the lungs, as we mentioned. They're contained within the thoracic cavity, except for what region of the thoracic cavity? What do we call the region of the thoracic cavity between the lungs?
The mediastinum.
There we go. It is indeed the mediastinum. Excellent. Notice the right lung consists of 3 lobes, a superior lobe, an inferior lobe, and a middle lobe. And notice there are 2 main fissures that divide it. We have an oblique fissure because it's at an angle, and a horizontal fissure, which is horizontal, dividing the superior from the inferior, and then the middle from the superior and the inferior. However, notice on the left side, there are only 2 lobes and only 1 fissure, an angled fissure, so an oblique fissure, that separates it into a superior lobe and an inferior lobe. Why the asymmetry? I know we already said it, but I'll ask it anyway. Why is there an asymmetry here? Why doesn't the right lobe look like the left lobe?
Was it to accommodate the heart?
Exactly. Remember, as we talked about, the heart is not on the midline of the body. It is offset slightly to the left. It also points laterally and anteriorly. So it actually projects even more left than it sits. We actually see that anatomically because notice there is an indentation in the left lobe that we don't see in the, in the left lung that we don't see in the right lung, known as the cardiac notch, where we have to be able to accommodate the heart. And that is something that is shown nicely on your model that we can see here from that virtual anatomy lab. So notice again, here we see there's that big, huge indentation to accommodate the apex of the heart, which is projecting left and projecting anteriorly. So because of that, there's only 2 lobes on the left side, whereas there's 3 lobes on the right. We looked at it on the pretty model. Here we see the same thing in the cadaver, where again, I think the thing that is most noticeable is again, this indentation that we see to accommodate the heart that is coming into this space. All right, as we mentioned, Our lungs are surrounded by a double layer of serous membrane known as the pleura. Visceral pleura is in direct contact with the organs. Parietal pleura lines the cavity. And in between, as someone mentioned, we have a thin line of serous fluid, just like we saw in the heart, that transudate. Notice also, and again, this was something we emphasized in 430. I'm not sure if we talked about it since then, but notice that each lung is individually shrink wrapped. So we have our visceral pleura here on what I'm going to assume is going to be the left lung, and the parietal pleura lining the cavity. Notice we can see where the two come together is that root where the blood vessels, the nerves, and the airways enter. The exact same thing is seen on the other side as well, where we have a visceral pleura, we have a parietal pleura, and again, the two come together at the root, and each lung is individually shrink wrapped, which is convenient because if you take an injury where fluid or air gets into this space, that can cause the lung to collapse, and we'll see why that occurs in the next class. And if that lung collapses and you're not able to move air into and out of your lungs, you could die. However, as long as not too much fluid gets in here and the other lung stays inflated, you'll be able to breathe while they rush you to the hospital and hopefully resolve the issue. So it is much more protective in its function by having each lung individually shrink wrapped. And again, it has that serous fluid inside this cavity. The goal is not to fill it, but to have a thin line, that transudate. And what does that transudate do?
Reduce friction.
Absolutely. One of the things that it will help to do is to reduce friction. But is that the only thing that it does? Well, I'm asking the question, so what's the obvious answer?
No.
No. So what else does it do?
Does it provide different pressure inside and outside the lungs?
Absolutely. One of the things that we want to do is we want to have what's going to be called a negative pressure inside here. We want the lungs to stay in contact with the thoracic cavity. So when the thoracic cavity moves out, the lungs move out with it. Because as we know, when you change the volume, you change the pressure and air can move. So one of the ways we're going to do that is by having pressure in there. The other way we do it is by having that serous fluid. Again, this is not the right time of year to be doing this, but if we were taking this class during the summer and we were all sitting out on our back porches where we have that glass coffee table and we have our nice big glass of ice cold lemonade that's sitting on that glass coffee table. And after an hour of me talking, I go to grab that glass to grab it and pull it off of that glass coffee table. What's going to happen?
There's going to be resistance.
Yeah, it sticks, right? The water gets in between the two and it holds the two together. Anybody know what mystical, magical property of water causes that to occur?
Surface tension.
Exactly. The other thing that we want here with this serous fluid is we actually want surface tension. We want the surface tension here so that the lungs stay in contact, stay stuck to the ribs, to the thoracic cavity. And so in this case, in that pleural cavity, yes, we want to reduce friction from the lungs constantly expanding and decreasing, but we also want that surface tension. We want it to stick the same way that that glass sticks to the glass coffee table. We want that surface tension holding it in place to help us to inflate the lungs. All right. There's really no muscle in the lungs itself. Right. So when we want to expand the lungs to get air into it, we need to expand the thoracic cavity and we want the lungs to move with it. Right. We can't contract the lungs by themselves. We squeeze the thoracic cavity and that squeezes the lungs and forces the air out. So we want that surface tension to help to hold this in place. Did that. All right, as I mentioned, we have two primary bronchi that enter into the 5. So let's do that. Two primary bronchi, because we have 2 lungs. That go into 5 secondary bronchi, or what we call the lobar bronchi, that supply the individual lobes. These then branch to form the tertiary bronchi. And these tertiary bronchi also are meaningful in what they bring air to. They supply individual regions of the lungs known as a bronchopulmonary segment. So in a particular lung, and I'll just do this really simply. Let's say that's a lobe of the lung. This lobe of the lung may be divided up into 4 bronchopulmonary segments. What's significant about this is each one of them has 1 tertiary bronchi that feeds air into it, but typically they also have 1 main artery that feeds into them and 1 main vein that feeds out of them. Again, this is very significant from a clinical standpoint, because when something like a lung cancer forms, typically those cancerous cells only form in 1 of these bronchopulmonary segments when it first forms. And if this whole compartment is only fed by 1 main blood vessel, fed out by 1 main vein, and has 1 main airway feeding into it, is it going to be really hard to cut that chunk out?
No.
No. And so when lung cancer is found quickly enough, then it is fairly easy to just remove that 1 bronchopulmonary segment with a very minimal surgery. So this compartmentalization of the lungs into these bronchopulmonary, and again, the name tells you everything about it. Broncho, referring to the bronchi, the airways. Pulmonary, referring to the blood vessels coming into and out of it for gas exchange. These bronchopulmonary segments compartmentalize the lungs into smaller portions. Now, the same way we have a different number of lobes on both sides, we have a different number of bronchopulmonary segments. On the right, and remind me again, how many lobes?
5.
3.
On the right, we have 3 lobes, and on the right, everybody in the class has 3 lobes, unless you had some kind of weird surgery, and everybody has 10 bronchopulmonary segments. So everybody's the same on the right. On the left is where you see variations. For starters, we all only have 2 lobes. But even those 2 lobes are compartmentalized different. individuals have somewhere between 6 and 10 pulmonary segments. So one person in this class may have 9, somebody else may have 6, someone else may have 10, someone else may have 8. From a functional standpoint, from an oxygen carrying capacity of the blood, from a movement of air into and out of the lungs, there doesn't seem to be any significant variations in people's functions based on this anatomical variation. Like I said, the only time this is really significant is when surgically one of these segments needs to be removed for some reason. But other than that, there doesn't seem to be any meaningful difference based on the number of bronchopulmonary segments you have. I love this picture from your textbook. What they have done, we'll talk about that in a second, is they have plasticined, basically poured plastic into a lungs and then peeled the lungs away from them. And they've color coded the individual parts. Specifically, we can see the trachea coming down, feeding into the 2 primary bronchi, feeding into the secondary bronchi. And then what they've color coded is the tertiary bronchi and all the branches that come off of them. So notice for this particular lungs, as we expect, there are 10 bronchopulmonary segments on the right side, 3 to the superior, 2 to the middle, and 5 to the inferior. And notice for this particular individual, there are 9 bronchopulmonary segments on the left, 4 in the superior and 5 in the inferior. But again, could someone else have 4 and 4 or 4 and 3? Or 5 and 5, yes. So over here, again, these numbers will vary per person, but the right side is always consistent. Now, notice the other thing that you can see. We go from primary to secondary to tertiary, and does the branching stop at that point?
No.
No, of course not. As you can see, in fact, there can be up to 24 levels of branching that occur. And actually, let's do this. Let's go to the whiteboard to write some of this out, because this is going to bring us to another important characteristic we want to talk about. All right, so let's start. One trachea leads into 2 primary bronchi. And again, these primary bronchi are 2 because we have 2 lungs. That leads to our secondary bronchi. And how many secondary bronchi do we have again?
5.
Why do we have 5?
To feed each lobe of the lung.
Yeah, 5 lobes, excellent. And that leads to our tertiary bronchi. And how many tertiary bronchi do we have?
$20.
Okay, 20 is the max, but does everybody have 20?
16 to 20.
There we go, 16 to 20. And of course, why do we have 16 to 20 tertiary bronchi? Because we have 16 to 20 what?
Bronchopulmonary segments.
Excellent. And then, like I said, from on and on, continues to divide up to 21 orders of division. Now, obviously, as they divide, they get smaller and they get more numerous, but there are some other distinctions that occur as well. As we mentioned, our trachea is lined with a ciliated, pseudostratified, a columnar epithelial tissue. And it turns out so do the primary bronchi. So both the trachea and the primary bronchi are lined with a ciliated pseudostratified columnar epithelial tissue. However, starting in the secondary bronchi and continuing into the tertiary, our tissue gradually, remember, when we looked in the esophagus, it was an abrupt change from the non-keratinized stratified squamous to the simple columnar. But here we get a gradual change to a simple columnar. And that simple columnar will travel all the way down till we get to the smallest bronchi that are known as bronchioles. And in the bronchioles, they are lined with a simple cuboidal epithelial tissue. Oops, that's not what I wanted. But there's one other big change that takes place as well. In our trachea, in our primary bronchi, remember we had those C-shaped hyaline cartilage rings. But as we start to get to the secondary and tertiary and smaller, those rings break up into irregular plates of hyaline cartilage. Until finally we get to the level of the smallest bronchi, and those bronchioles have no hyaline cartilage on them as well. And one last thing about these secondary and tertiary bronchi. Not surprisingly, these secondary and tertiary bronchi have a thicker smooth muscle wall than the primary and the trachea. And why do you think that would be the case? The primary bronchi and the trachea are bigger, so shouldn't they have a bigger smooth muscle wall? Come on, guys, I'm not just talking to hear myself talk. What do you think? Well, if you have a big, large C-shaped chunk of cartilage holding you open, are you going to be able to change the diameter of that very easily?
No.
No. So there's not much point in having a big, huge, thick, smooth muscle wall. However, if now you're just held together by a bunch of irregular shaped plates, now the smooth muscle can have much more effect on the diameter. And so it is typically at the tertiary or even smaller that our sympathetic and parasympathetic are going to have the biggest effects on the movement of air, because we're able to more easily dilate or constrict those airways because they don't have that rigid ring holding them in place. Notice if we go back now to the lecture, we can see these things that we were talking about. Notice when we start in our trachea, in our primary bronchi, again, there's still that ciliated pseudostratified columnar epithelial tissue. There are still those rings of cartilage. But notice as we start getting to the tertiary and the secondary and tertiary, those rings start breaking up into irregular plates of cartilage that go away when we reach the bronchioles, which have no cartilage on them. Our epithelial tissue gradually changes to simple columnar until it gets to the bronchioles, where it is simple cuboidal. And just out of curiosity, what do you think the epithelial tissue in the alveoli is?
Simple squamous.
Simple squamous, where we want that rapid gas exchange to take place. And now that we have these irregular plates of cartilage, it is easier to change the diameter of these airways to direct the air where we want it to go. So again, we will see an increase in the smooth muscle in these regions as well. All right. Questions on that? All right, excellent. With that, that is going to bring us down to the level of the bronchioles, and we still need to talk about what's going on down here. But clearly, we are in need of another break. So during this break, you are required to go and drink some type of stimulant, tea, coffee, Red Bull, something, do a bump of cocaine, do something so that you guys are a little bit more interactive for the second half of this. Well, not second half, but for the last portion of this lecture. Don't do cocaine, but do any of those other stimulants, assuming you have the heart that's doing it, or just, heck, be more interactive when we come back. Again, remember, I could easily be asleep right now and you guys could be watching old videos. I am here interacting with you to make this a better lecture for you. So make it a better lecture for me by interacting. So let's take a 15-minute break. It's 2:31. That means coming back at 2:46. And we will restart and I will start the recording at that time. All right. Any questions? All right. I will see you guys in 15 minutes. All right. Any questions before we get started? All right. So we started out here in space. We have been following the air as it would pass through our conducting zone, going from the nasal cavity, through the pharynx, through the larynx, trachea, into the bronchi, all the way down to the bronchioles, till we finally get down to the distal portion of this airway, where our conducting zone ends. And our conducting zone ends, appropriately enough, at a structure known as the terminal bronchiole. Now, again, one of the important things to remember, basically, there's our cut right there, is the function of the conducting zone. Remember, the conducting zone is all about moving the air, but more important than moving the air, remember, it is also about processing the air. And by processing the air, I mean cleaning it, I mean warming it, I mean moistening it. All right. Once we get to the terminal bronchiole, notice we're not at the last bronchiole. There is another bronchiole right here. However, this bronchiole, known as the respiratory bronchiole, is different from the terminal bronchiole because our respiratory bronchiole has an alveolus on it. Now, notice this one respiratory bronchiole has one alveolus on it. Is that going to play, that one alveolus going to play a massive, massive role in the entire gas exchange that is going on inside of your body?
No.
No, of course not. But the fact that it has at least one bronchiole on it does mean that it will participate in gas exchange. And so any structure that participates in gas exchange, no matter how small, is no longer part of the conducting zone, and as we talked about now, is part of that respiratory zone where the gas exchange takes place. So here, this terminal bronchiole is the last location. Notice we're down here, and as we look at it, we can see it has no cartilage on its outer surface. It has some irregular smooth muscle banding around it. And remember, as we talked about, the epithelial tissue lining these are going to be simple cuboidal. So that is what we've got going on here. Now, these terminal bronchioles feed into these compartmentalized structures known as a pulmonary lobule. A lobule, again, is a term we've heard before. And notice, like we saw lobules in the liver, like we've seen lobules in the glands of the endocrine system, a lobule is basically defined by the structures that are compartmentalized by connective tissue. So there is a connective tissue septum that wraps around these structures, and these are basically structural and functional units that we see here in these pulmonary lobules. We have a nice one here on this illustration, and the other place we have a nice view of one of these is on a model that's in the classroom. Here's one of these models, an older model that we can see from the posterior side, and this is a better view from the anterior side that we can see this. And there's a newer model that we have that sees all of these things as well. So again, notice basically we would get to that terminal bronchiole right about here, and then this would be our lobule. Notice this lobule contains these grape-like clusters. These grape-like clusters are what are known as alveolar sacs. And again, here from here down takes place in gas exchange, part of our respiratory zone. Notice here we can see one of these alveolar sacs that have been cut open. Here we see it as well in a histology slide. And again, our lung is filled. And again, they made up this number. I didn't do the counting, but I'll take their word for it because I'm not going to check their work. Each lung contains over 300 million alveoli. Big, huge air-filled spaces. If you get the opportunity to go to Open Lab in the classroom, in the back of the classroom in 418 is actually a big chunk of what feels like styrofoam, which is actually a freeze-dried lung. And when you look at it and you loft it, like styrofoam, it is incredibly light and incredibly porous, because as you can see from the light microscopy, alveoli are thin, simple squamous epithelial tissues filled with air. So it's basically mostly space, and a whole lot of space, somewhere between 70 and 140 square meters. And like I said, to put that in perspective, it's about the size of a small tennis court, all folded up inside of your thoracic cavity. Here, as I mentioned, we see that alveolar sac. The sac is the entire cluster of grapes. And like when you look at a cluster of grapes, all you see are the grapes, like all you see are the alveoli. However, is it just hope that is holding those grapes together?
No.
No, of course not. There is a stem that is deep to all of those grapes holding them together. And that's exactly what we see here. Our alveolar sac is held together by an alveolar duct. That alveolar duct is basically just a conduit, just a passageway. In fact, if we look back at the previous picture, we can see that the alveolar duct is really just the space. Notice we can see a couple things here when we look at the light microscopy. Notice I have the little bit of bandings of muscles, a thicker wall that we see here. This is the bronchiole. Notice they've got this labeled as a respiratory bronchiole. How do we know this is a respiratory bronchiole? They're guessing? What's the difference between a respiratory bronchiole and a terminal bronchiole?
It has to do with gas exchange.
Yeah, notice this particular bronchiole actually has an alveolus on it. And so the fact that we see this alveolus means that it must be participating in gas exchange to some extent. And that's what makes it a respiratory bronchiole. Notice we can clearly see the wall of it. It's very defined in its shape and structure, which is almost the opposite of our alveolar duct. Notice our alveolar duct is really more of a space on the slide that we can see all of the alveoli coming off of. It's kind of like, I think of it kind of like the hallway of a hotel. Right? If you walk down a hallway in a hotel, all you have is a bunch of doors that open up into a bunch of rooms. And that's basically what we have here. We have all these rooms coming off of this single hallway. And so that's kind of what we've got here. At the center of our sac is this alveolar duct, which of course is made by a cluster of alveoli. And of course, alveoli is the plural. Alveolus is the singular. All right, so fun with vocabulary. Questions on that?
Yeah, is the duct just a continuation of the bronchiole?
Yes, so the respiratory bronchiole does continue down into the alveolar duct, but notice the anatomy of it changes. Unlike the bronchiole, it is not lined with any kind of smooth muscle. Right, it basically is just a little bit of structure formed by a simple cuboidal epithelial tissue hallway. And so notice on the slide, it's actually the alveolar duct is actually more space than it is anything else. It doesn't have a tremendous amount of structure. It's just that hallway connecting all of the rooms together. All right. Whereas the bronchioles will have a much more distinct wall and will have, like I said, will have that smooth muscle that makes it very obvious as well. So we can probably see that pretty distinctly and differently when we look at that. We did all of that. Excellent. We did all of that. If we take a microscopic view of an alveolus, we can see some of the anatomy. Notice here, using an electron microscope, we can see the ball-shaped structures that are the alveoli. And notice over the top of them, we can see all of the twisted and turning strands that are the pulmonary capillaries. So this mesh-like structure that sits over the top is the pulmonary capillaries. So notice if we were to take a cut through this structure and look at it from the side, that's what we would see here. Notice as we look at this illustration, we can see the branches of the pulmonary capillaries that are wrapping around that ball-shaped alveolus. And we can see the cells that are making up and found inside of this ball-shaped alveolus. Notice the primary cells, as we've kind of already hinted at, are these beige, broad, flat, squamous cells known as the type 1 pneumocytes. So all of these broad, flat cells, these tiles, here we see one, here we see one, here we see one, forming the majority of the wall of our alveolus are what are known as type 1 pneumocytes. And their function is structure. They form the structure, and they're going to form part of that respiratory membrane where our gas exchange, whoa, that's way too big, where our gas exchange will take place between the alveolus and the pulmonary capillary, or between the air and the blood, as we've talked about. However, notice if you look closely, while these simple squamous type 1 pneumocytes make up the majority of the wall of the alveolus, there are these other cells that are embedded within the wall of the alveolus as well. And these are what are known as the type 2 pneumocytes, or what are also referred to as septal cells. Septal cells produce a substance known as surfactant. Anyone know what surfactant is used for? Well, let's think about what we were talking about in the pleural cavity. In the pleural cavity, we have a thin layer of serous fluid that provides surface tension, so that when the thoracic cavity expands, the lungs stay stuck up against it and move in relation to it. When you get a balloon and pull it out of the pack, what is the first thing you do to it?
Stretch it
Yeah. Why do you stretch it?
to make it easier to blow up.
True. But why does stretching it make it easier to blow up? You're 100% correct. But do you know why?
To remove some of its elasticity.
So, or to help to increase the elasticity by stretching it out, it may help with the elasticity. That may help it to not pop when you're blowing it up, but it's not actually what allows you to get the air in there to begin with. If you first take a balloon out of the bag and try blowing into it right away, it is incredibly hard to blow up, to get air into it. Not because it hasn't been stretched out yet, but because the two sides are stuck together. When you stretch it, you break the two sides apart from each other. And when you break the two sides apart from each other, it's much easier to get the air in there. Well, when I breathe out and air leaves this alveolus, a huge moist environment, do I want the two moist sides of that alveolus to come together, to be attracted to each other by that surface area and collapse entirely? So that next time I have to force enough air in there to push them apart to fill my alveolus so gas exchange can take place?
No.
No, that would not be an efficient way to do things. And so what this surfactant actually does is it breaks up the surface tension. Surface tension outside of the lungs, good. Surface tension inside of the lungs, bad. We don't want the two sides of the alveolus to collapse and stick together. So by producing surfactant with these type 2 pneumocytes, these septal cells, it breaks up that surface tension and allows the alveolus to stay opened easier. Not surprisingly, one of the things that happen as we age is our type 2 pneumocytes produce less surfactant. So as we age and we're producing less surfactant, what happens to more of our alveoli?
They lose their shape.
Yeah, they lose their shape and they collapse. And then is it going to be as easy to inflate our lungs?
No.
No, so we can't get as much air into our lungs. Our gas exchange isn't going to be as efficient, right? And our respiration rate goes up, our lung volume goes down, right? All of these things. Yes, there's also the fact that you worked in a coal mine for 20 years and you smoked 2 packs of cigarettes a day. Those things also damage and decrease the volume and the functionality of your lungs, but sometimes just getting old decreases those things as well.
So is oh, I'm sorry.
Go ahead.
So is surfactant made of lipids or mucus?
It is not mucus. It's a great question. I don't actually know what the substance is to break up the surface tension. It wouldn't be lipids because they would separate from the water, so that wouldn't work that way. It would have to be something that was water soluble because basically the physics of it is surface tension is caused by the hydrogen bonds of the hydrogens and the oxygens being attracted to each other. Those hydrogen bonds are what allows a droplet of water to pool on your window sill. It's why it hurts when you do the belly flop from the high dive, all that surface tension. It's why bugs can skitter along the surface of the water. So to break up those hydrogen bonds, it would have to be something that was water soluble, but probably something that had some polarity to it to break up the surface tension. So I don't actually know what the substance is. That's a great question. I don't have an answer for it. Probably something like, what's an antifreeze? Anybody know what's an antifreeze? Antifreeze is glycerol. So it's probably something sugar-based. So maybe a sugar or alcohol-based, a water-soluble alcohol, something like that. Those would be my guesses. All righty. So those are the cells that make up the walls of the alveoli. But notice there are also these blue cells that are in here as well. These blue cells are actually not a part of the wall. They actually migrate from alveolus to alveolus. And these are known as the alveolar macrophages, or what are also known as dust cells. And guess what they do?
Do they trap dust?
Yeah, exactly. Any dust and debris that may get here, pathogens, other microscopic things that are in here, they're going to help to break down, recycle, and destroy any components that could be potentially harmful in the lungs. All righty. So those are the cells that make up our alveolus and are found inside of our alveolus. Questions on that? All right, excellent. Here's another view of one of these alveolar sacs. And again, these pulmonary capillaries that wrap around it. These form, as we mentioned, our respiratory zone or our respiratory membrane, where our gas exchange takes place. And again, that gas exchange is taking place between the air and our blood. And we don't want to necessarily let anything that is in the air into our blood. So it's also known as the air-blood barrier. And if we think about it, we now know all of the anatomy that is going to make this up. What is the primary cell of the alveolus, forms the wall of the alveolus?
Simple squamous.
Yeah, we have a simple squamous epithelial tissue. All right, it's that type 1 pneumocyte. And it is a simple squamous cell. A single flat cell. Whoops, hold on. Let's I wrote that in red. Let's do this in red. A single flat cell. And again, like most simple squamous cells, often the nucleus bulges out on the wall of it. Excellent. We also have the wall of the pulmonary capillary. What type of cell forms the wall of the pulmonary capillary?
Type 2 pneumocyte? Or,
Well, but again, that's the alveolus. Think back to the cardiovascular system. The cardiovascular system, what type of cell forms the wall of a capillary?
Simple squamous epithelial tissue.
Yeah, exactly. Remember, it's the tunica and intima. We have that simple squamous epithelial tissue, right? What we would call the endothelium. Remember, the endothelium is continuous with the endocardium on the inside of our cell. And as we mentioned, it is just, again, a simple squamous epithelial tissue with a bulging nucleus that's sticking out. And like all epithelial tissues, they have an apical and a basal surface. And what's on the basal surface of an epithelial tissue?
Connective tissue.
Not all epithelial tissues sit on top of connective tissues, but remember when we way back in 430, when we talked about epithelial tissues, remember on the basal surface, we talked about there being a glue that holds them to the tissue underneath. Does anybody remember what that glue is that holds the epithelial tissue, lines the basal surface of an epithelial tissue, holding it to the tissue underneath?
basal lamina?
Basal lamina or basement membrane. Right? Each of these have this. So basically it is the fused basal lamina or the fused basement membranes of these two simple squamous epithelial tissues that form our respiratory membrane. Again, remember, gas exchange is passive. So we want to make it as simple as possible. So we want to have it as short of a distance to travel as possible. And does it get much shorter than two simple squamous epithelial tissues with a little bit of glue holding them together? No, that's about as simple and as basic as it gets. We have that simple squamous endothelium of the capillary, that simple squamous epithelium of the alveolus, that type 1 pneumocyte. And then their fused basement membranes, their fused basal lamina. And like I said, this is about as short of a distance to travel as possible. In fact, all a gas has to do to get from one side of this to another is go about half a micron. Which, if you think about it, kind of makes sense why this is happening inside of your chest. Obviously, we have a huge surface area on the outside of our body where gas exchange could take place. But do we want this boundary between us and the outside world here on my shoulder to be half a micron in width?
No.
No. So that's why we have these tucked and folded way deep inside of our thoracic cavity with all that protective conducting zone to filter and clean and moisten the air to make the movement as efficient as possible. All right. Questions on that? All right. With that, we are almost done with our anatomy, except for one major modification we need to make. Our lungs actually have two circulations associated with them. Let's cheat and go to our whiteboard to do this. All right. Again, you guys all know what an amazing artist I am when it comes to drawing the heart. So there you go. Hold on, I'm not done. Perfect. As you know, this is my incredibly amazing interpretation of what the heart looks like. Almost completely accurate. And remind me again what these four chambers are. What would this one over here be?
Right atrium.
Excellent. And beneath it?
Right ventricle.
And across from that?
Left ventricle.
And above that?
Left atrium.
Excellent. Now, as we have talked about already way back in for the beginning of this section, the first section of 431, we know that there is a pulmonary circulation. Describe that pulmonary circulation for me. What does it do? Let's start first with what its job is. It carries blood where to where to where?
From the body to the heart, to the lungs, and back to the heart.
Okay, well, we don't have to put body in there, but you have the right idea. From the heart to the lungs and then back to the heart. Right? Why?
To oxygenate the blood through the lungs.
True, although now we know a little bit more gas exchange. Part of it is to get oxygen into the blood, but also to get rid of CO2, which also reminds us of the condition of the blood. We know the condition of the blood, as you mentioned, is going to be oxygen poor. No, that was right. But what do you think about its CO2 levels?
CO2 rich.
Yeah. Or high levels of CO2. So low levels of oxygen, high levels of CO2, or oxygen poor, CO2 rich. Excellent. And describe for me the pathway. Where does the pathway of this pulmonary circulation begin?
The right ventricle.
Excellent. So we have that oxygen poor blood leaving the right ventricle. Out of what blood vessel again?
Pulmonary trunk.
Pulmonary trunk. And it goes from there where?
Left and right pulmonary arteries.
Excellent. So the pulmonary arteries. And then eventually it goes where?
To the lungs.
Absolutely, to the lungs, to the pulmonary capillary, where our gas exchange is going to take place. So let's throw some labels on here real quick. And of course, here in the pulmonary capillary, our external gas exchange takes place. And remind me again, which way does oxygen move?
From the tissue to the blood. From the
Well,
blood to the tissue.
Okay, close. Here we've got external gas exchange taking place though. So that's between blood and what other substance?
Interstitial fluid.
Okay, let's think of the structure. Our pulmonary capillary is exchanging blood with what structure?
Air.
Well, the structure would be an alveolus, but absolutely, you've got the right idea. Oops. Excellent. So oxygen is going which way? From air to blood or blood to air?
Air to blood.
Excellent. And carbon dioxide?
From blood to air.
Excellent. We then have our oxygen-rich blood, which goes back where?
Left atrium.
Via what? Pulmonary capillaries feed into our
Pulmonary vein.
pulmonary veins. Excellent. Which feed back into the left atrium. Perfect. All right. Actually, no new information here. This is stuff we already learned in this class. All right. Oh. Right, with the exception of thinking of which way the air is going, all of this is structures we have talked about before. So we comfortable with that? Hopefully.
Yeah.
Okay, excellent. However, what's interesting is that there is actually a second circulation. And this second circulation is the bronchiole, not bronchiole. Remember, bronchioles are tiny. This is the bronchiole circulation. And based on its name, guess where it goes? It's not a trick question. Where do you think a bronchial blood vessel goes?
To the bronchioles.
Yeah, to the bronchi, absolutely. This goes to the bronchi and the other, the smooth muscle and other connective tissues of the lungs. Basically, everything but the alveoli. Now, why might we want blood going to the smooth muscle, going to the bronchi, going to the connective tissues of the lungs?
They need oxygen to function.
Yeah, they're living tissues. Those tissues need oxygen and nutrients to do their work. They need to get rid of wastes. That doesn't sound a whole lot like a pulmonary circulation to me. That sounds like what we were talking about before with the systemic circulation. And that's what this actually is. This is a branch of the systemic circulation. Based on that, what's its pathway going to be?
It will start from left ventricle to the aorta.
Excellent. I will use pink to emphasize the difference. it is going to come out the left ventricle. Oh, wait, that's way too small. Out the left ventricle. All right, into what?
Ascending aorta.
From there, where do you think it goes?
Aorta.
Or
The
aortic
aorta.
arch.
Yeah.
Into the thoracic aorta. And from there, in our thoracic cavity, there are bronchial arteries that then branch off. And as they branch off, they are going to go to the tissues, the rest of the lungs. So if we cheat and draw our cube here, that is the lungs, this is going to go to the connective tissue septums and the smooth muscle and the bronchi, basically everything but the alveolus. And here, it's going to feed into a systemic capillary. And in the systemic capillary, what type of gas exchange takes place?
The oxygen will move out of the blood and CO2 will enter the blood.
Excellent. From the tissues. So would that be internal or external gas exchange?
Is it internal?
Yeah. Where you are correct. Oxygen goes from the blood into the interstitial fluid. And CO2 goes from the interstitial fluid into the blood. So again, we're going to have a capillary here. So I'm going to feed into a capillary bed here where this exchange is going to take place. Between the tissues. This of the force is then going to feed into what structure? What type of structure do you think would come out of this then?
Venule.
Yeah, you'd have a venule that would feed into what?
Vein.
Right. And what do you think, how you think we would identify this vein? How about the bronchial vein? And where would that bronchial vein go?
Eventually the inferior vena cava.
Which would then feed it into which chamber of the heart?
The right atrium.
Right atrium. And that would make a lot of sense, except for one fact. Turns out bronchial veins are very lazy. Here they are sitting here with all this blood that needs to go back to the heart. And they say, lo and behold, hey, look, here's some pulmonary veins that are already going back to the heart. Deal me a solid, dude. I can't be bothered to go all the way back to the heart by myself. Why don't you carry my blood back for me? So these bronchial veins actually feed into the pulmonary veins. So notice two circulations come out, but only one goes back in to the heart. Now, is that going to have any effect on the blood? Well, I'm asking the question, so what's the obvious answer?
Yes.
Yeah,
Is it going to make the blood more CO2 rich?
yeah. So that bronchial vein feeding into the pulmonary veins will change the condition and it turns out not so much that it makes it more CO2 rich, but it does end up dropping the oxygen levels in the blood slightly. So basically, when it leaves the alveolus from the pulmonary capillary, it's very, very oxygen rich. And by the time it gets back to the heart and it's mixed with the pulmonary vein blood, it's just very oxygen rich. And we can actually put numbers to this. We'll worry about the numbers later, but when we talk about gases, we talk about them in millimeters of mercury. When it leaves the pulmonary capillary, it is at 104 millimeters of mercury. So when it leaves the pulmonary capillary. When it reaches the left atrium, it's down to 100 millimeters of mercury. So notice it's not a huge change, just a tiny little drop. So the pulmonary circulation is definitely something we talked about at the beginning of this class, but we hadn't talked about that bronchial circulation. And so that is something that we needed to talk about here. Here, I've got this amazing picture that does a great job of representing it. But again, your book does a good job of describing this too. Pulmonary arteries, again, come off of the right ventricle with oxygen poor blood going to the lungs, feeding the pulmonary capillaries for that gas exchange to take place, and come back via the pulmonary veins that go into the left atrium, where it is now oxygen rich and has less CO2 in it. Our bronchial arteries, though, are systemic blood vessels. They come off of the thoracic aorta. Now, like the pulmonary and like the bronchi themselves, all of these still enter the lung in the root. And their job is basically to supply the lung tissue to everything except the alveoli. And because they're lazy, they can't be bothered to carry the blood back to the heart themselves. So basically, they anastomose with the pulmonary veins to carry them back. So it is the pulmonary veins that carry all the blood back to the heart. Now, as I showed you, there is this model in the classroom that shows this lobule of the lung, and it is an almost perfect model. Let's take a look down here at the distal portion of it. Notice as we look at this distal portion of it, there's a couple of key things that we're able to see. Notice there are these, and I'll make them bigger, blue blood vessels that follow along the alveolar ducts and the bronchioles and feed into all of these capillaries. What do you think this blue blood vessel I've just labeled is?
Are they the pulmonary arteries?
Yep. How do you know?
Because they're feeding into the alveoli.
Yep. And what color are they?
Blue.
Which indicates what?
They're oxygen poor blood.
Exactly. Excellent.
Oxygen poor blood.
So then this red circle, what would that represent?
Alveoli.
True, but the red dots, the red lines, what do those specifically represent?
Capillary bed.
Yeah, and not just any capillary, but the pulmonary capillary bed, which, notice, feed into these red blood vessels that, again, no longer worry about following the alveoli because they've gotten what they need, and they're on their way back towards the heart. What do you think this nice dark red blood vessel indicates?
Pulmonary veins.
There you go. Carrying that oxygen-rich blood back to the heart. However, notice there is a second red blood vessel. This second red blood vessel is much lighter in color, but still red. But notice it is not following the alveoli. Instead, it is located out here in the connective tissue septum of my lobule, coming down to the connective tissue wall on the outside of the heart, where we see the serous membrane with our simple squamous epithelial tissue lining the outer surface. Guess what this blood vessel is?
Bronchial arteries.
There you go. Notice it feeds into a capillary out here in the connective tissue. What kind of capillary is this one? Well, it's not a pulmonary capillary, so what other choice do we have?
Bronchial capillaries.
True, that I would accept that, but remember, we said the bronchial circulation is part of the systemic circulation. So this is just simply a systemic capillary where internal gas exchange takes place, right? Remember, external gas exchange takes place in a pulmonary capillary. Internal gas exchange takes place in a systemic capillary. And notice finally, you can see that the vein feeding out of this goes right into that pulmonary vein carrying the oxygen-rich blood back. So notice we see all of these bronchial arteries and veins all feeding into the pulmonary veins, carrying the blood back. So notice we see all of our blood vessels are two different types of capillaries, their relationship with each other. Remember, as I mentioned, this is an almost perfect model. Of course, what's the key word in that statement?
Almost.
Almost. So there is a big problem, which I'll show you in just a minute, but let's make sure we understand this first. Spending all the time on it that I am, are you likely to see this model on an exam? Do you need to know this anatomy?
Yes.
Yeah.
Yeah, absolutely. So any questions on this before I show you the one minor major error this model has?
Professor, so the bronchial circulation starts in the left ventricle and ends in right.
No.
Left atrium.
Yes. It ends, so it starts in the left ventricle and it technically would end in the left atrium, but really it ends when it feeds into the pulmonary veins.
Okay. Mhm.
So it starts at the left ventricle and ends at the pulmonary veins. But you're right, the blood ends up back into the left atrium. Great question. Any others? All right, then let's look at the problem. It's up here at the top. Notice here at the top, as we saw, we have our pulmonary arteries that are coming and feeding into the pulmonary capillaries, and our pulmonary vein, which is taking, let's make it pink so it'll stand out more. That pulmonary vein, which is carrying the blood out of the capillaries into the connective tissue and out of the lungs. All right? Yes?
Yes.
Yes. However, remember I also told you that the pulmonary arteries follow the bronchi, follow the alveolar ducts, follow all of those things down. And if you notice, they've got it offset to the side. This should actually be wrapped around the bronchi, and the pulmonary vein should be offset over here to the side. So the top part of this, if we cut this off and reversed it, then it'd be perfect. So the top part of this can be confusing for some students because they know that the artery is supposed to wrap around the bronchi and the vein is not, but they have these in the wrong position. So the colors are correct. Everything else is correct. Everything's perfect. This should just be wrapped around that, and this should be offset. So other than that major minor problem, it's not an issue. But notice also, as we did before, if you just look at the bottom, then the problem's gone. So basically, if we just cut the top of this model off, which I have requested numerous times, but they won't do. But yeah, if we just cut this model off right here at the top like this, then it'd be perfect. But nobody else wants to do that. So as long as you ignore the top, you'll be fine.
Professor, so the color indicates oxygen poor or oxygen rich. It doesn't indicate artery or vein.
Remember that that was one of the main points we made in our cardiovascular system, that most arteries are oxygen rich, most veins are oxygen poor, but remember, most is not all. So remember, we couldn't use oxygen carrying content as a way of identifying arteries and veins. And the reason for that is the pulmonary circulation. The pulmonary circulation is what screws it up for everybody else. The pulmonary artery is oxygen poor. The pulmonary vein is oxygen rich. They're the ones that are backwards, but remember why they're backwards. We're taking the oxygen poor blood away from the heart to the lungs to get oxygen. And then once we get the oxygen, we have to take that oxygen back to the lungs to spread it through the rest of the body. So these two blood vessels are the pulmonary blood vessels, and they're the ones that screw it up for everybody else. Notice, as we talked about, the bronchial artery is a systemic artery, and like all systemic arteries, it's oxygen rich. So here we have an oxygen rich artery, but here we have an oxygen poor artery. And here we have an oxygen rich vein. Oh, that should be A. And it is because the pulmonary circulation screws it up for everybody else. So remember, oxygen carrying capacity is not how we can identify arteries and veins. The way we're able to identify these three, and again, let's look at the bottom because then we don't have to worry about the issue, is where they're located and where they're going and what their job is. This is an oxygen poor blood vessel following the pathways to the capillaries.
Not kidding, huh? Huh?
Yeah, exciting stuff.
No, if you want to do it, be my guest.
I agree. All right, into a capillary, pulmonary capillary, where the gas exchange takes place, gets oxygen rich, and then that oxygen rich vein carries the blood away from the pulmonary capillaries and out to the connective tissue and back to the heart. So these are the pulmonary circulation. However, as we also talked about out here in the connective tissue, we have that systemic artery, that bronchial artery, coming to a systemic capillary where internal systemic gas exchange takes place, and then it feeds into that pulmonary vein. All right. Questions on that? All righty. If we understand the differences in these two gas exchange, blood supplies and the gas exchange that takes place in them, then that is everything that we needed to talk about for today. What we will pick up with on Thursday is we will start the processes, starting first with pulmonary ventilation, and then from there we will go into gas exchange. Now, as you know, I'm not a huge numbers person. Like I said, numbers are things that usually can be looked up, but occasionally we have numbers that are important. And these gas exchange numbers are going to be important. These are numbers. So if you recall, where did it go? For instance, these two numbers right here, 104 and 100. These are going to be numbers that are going to be important, but we're going to talk about the condition of the blood as it leaves the heart, as it comes back to the heart, as it's taking place in a external gas exchange in a pulmonary capillary, internal gas exchange in a systemic capillary. We will talk about all of the pressures of the gases, because remember, gas exchange is a passive process. And the same way glucose moves down a concentration gradient, oxygen moves down a concentration gradient. CO2 moves down a concentration gradient. But when we talk about gases, we don't talk about a concentration gradient. Instead, they call it a pressure gradient. But it's the same thing. Wherever there's more oxygen, oxygen is going to move away from that. Wherever there's more CO2, it's going to move to where there's less CO2. So it's the exact same type of concept. And so that's what we need to talk about next. All righty, questions on that?